Thank you for the introduction, Marina, and thanks to Marina and Sasha and everyone else for inviting me to come. It's been an interesting visit. This is my first visit at a pharmacy department, a division of pharmacy. So it's been really fun meeting the faculty here and the students and talking about research, especially research related to nanoparticles. Now, my group has been working on magnetic nanoparticles for quite some time. And I am a chemical engineer by training and a fluid mechanician. So we're really interested in fluid physics and the colloidal hydrodynamics of nanoparticles in suspension. And we're also interested in biomedical applications that are related to that. Before I start, this is maybe in this environment, you guys are all aware of things like this, but I want to start by saying that the biological environment is complex and it is nano. And I like, there's these watercolor images by David Goodsell, which I find very pretty, but also very informative. He has a really nice book that you can read, and it's filled with pictures such as this. This is an image of a cross-section of an E. coli bacterium. The flagellum here is about 20 nanometers in diameter. This one is of an HIV particle in blood serum. The HIV particle is about 120 nanometers in diameter. And this one is of a red blood vessel, a red blood cell in plasma. And the interesting thing here, the hemoglobin protein molecules are about 7 nanometers hydrodynamic diameter. So two things stand out. One is that a lot of biological structures are nanoscale in size. And two, that a simple fluid, deionized water, is never a good model for a biological medium. And I point this out because a lot of research, I think, being done to develop new nanomaterials is limited to characterization in the lab to their behavior, such as colloidal stability, in extremely simplified fluids that are not representative of the actual environment that these nanomaterials are going to encounter. To put the sizes in perspective, these would be illustrations of typical particle sizes. This would be a 20 nanometer particle, a 60 nanometer particle, a 200 nanometer particle. And I've tried to make this such that it's sort of to scale. And the idea is to really drive home the fact that these particles, A, have similar length scales as the structures in the biological systems, but also, for example, for this particle, the idea that the plasma, for example, is a continuum, a simple fluid of simple properties such as a single viscosity, et cetera, is probably not completely applicable. So we have to take these things into account. And the only point I want to make is that, in my opinion, engineering future applications of nanoparticles, or in my case, magnetic nanoparticles, in the biological environment really requires an improved fundamental understanding of nanoparticle dynamics in complex, crowded, and confined environments that are representative of the biological medium. And I really enjoyed my conversation with Sam. I don't know if he's around, but I really enjoyed my conversation with Sam earlier today because I think he's looking at some of those things too, and I think more people need to be looking at that. Okay, so magnetic nanoparticle suspensions have been around for a long time. These were developed in the 1960s and are commonly called ferrofluids. These are colloidal dispersions of magnetic particles that undergo rotational and translational baryon motion. And they have a series of applications that are established commercially. More recently, because of their small size and because most ferrofluids consist of iron oxide particles, which are believed to be biocompatible, there's been a lot of interest in their potential biomedical applications. And the reason these materials are interesting in biomedical applications and in many applications is simply because you can apply a magnetic field and you can manipulate them, you can move them, you can exert forces on the particles, you can make the particles rotate, and there are certain things that you can do with this. Perhaps the best known biomedical application of iron oxide or magnetic nanoparticles is the idea of using them for magnetic resonance imaging, which is approved in the United States and in Europe for use in humans as negative MRI contrast agents. And the important thing about this, the idea that they've been accepted, they can be used clinically, is that at least for the particles that went through this trial, which consisted of iron oxide particles coated with a polysaccharide dextran, we believe that there are no major toxicity issues, okay? Now, acceptance as MRI contrast agents has been harder, and actually the product was pulled off market in the United States, not because of a toxicity issue, but more of a lack of acceptance of the idea of using a negative contrast agent. but the principle is still there. I think more exciting are other applications that take advantage of the fact that you can exert forces and torques, and you can dissipate energy using magnetic particles. And some examples are the idea of magnetic drug targeting, where you would associate a drug to a magnetic particle, and you would use an externally applied magnetic field, specifically a magnetic field gradient, to exert forces and concentrate the particles in a targeted tissue, thereby aiding accumulation of your drug in the intended tissue. Another idea is the actuation of ion channels and cell surface receptors. And here what you want to do is you want to modify the particle such that it attaches to an ion channel or a cell surface receptor that is mechanoresponsive. And then you apply a magnetic field to either pull on the particle or twist the particle, and that can induce a response. There's been a lot of work, and actually close to here, Ben Yellen has been working or has worked in the past on using magnetic nanoparticle suspensions to promote assembly of cells. There's also work on nanomagnetic gene transfection, the idea of loading particles with genes and then using magnetic forces to pull or push the particles into a cell, thereby enhancing delivery of genetic material into the cytoplasm. And then the last one that I'm going to get back to in the second half of the talk is the idea of treating cancer, let's say, by using magnetic particles to deliver thermal energy into a tumor. So before I go on, the applications that I'm going to talk to you today take advantage of the response of magnetic particles to time-varying magnetic fields. And I want to point out that there are two ways in which particles respond to a magnetic field. One is you have a particle, it has a magnetic dipole shown in green, and you apply a magnetic field in a certain direction, and the particle physically rotates so that the dipole aligns with the magnetic field. And this is what's called Brownian relaxation. The other possibility is that you have a particle, and you apply the field, and the dipole physically, rotates within the particle without physical rotation of the particle itself. And this is what's called Niel relaxation. As you might imagine, the response, the dependence of each of these two mechanisms on magnetic properties of the material and on the surroundings are going to be different. And this can be quantified, let's say, by these expressions for the characteristic relaxation time, which is the time it takes for the particles to align with the new field, for the Brownian and the Niel mechanism. And what I want to point out is that in the case of Brownian particles, that is particles that are magnetically blocked at room temperature, the relaxation time depends on the hydrodynamic diameter of the particle and the viscosity of the surrounding fluid, or more generally, on the rotational hydrodynamic drag on the particle. And in the Niel mechanism, the time depends more on something called the magnetocrystalline anisotropy constant. And the reason I'm pointing that out is, one, it's going to be important in some of the things that I'm going to work on, but it also illustrates the importance of knowing what you're doing, obtaining particles with known properties, obtaining particles with narrow size distributions. And I'm not going to go into the details, but the point is that if you have polydispersity in particle size, if you have aggregation, you most likely are going to have a combination of the two mechanisms of energy, of relaxation or response to magnetic field. So in the first application I'm going to show you, we want to use particles that physically rotate. And we want to talk about using the particles as nanoscale probes in complex fluids. And the work I'm going to show you are non-biological fluids, but like some conversations today or yesterday, we're interested in translating this into biological fluids. So the first thing is, what is this idea of nanoviscosity or nanoscale viscosity, or why does it matter the size of the probe that you're using? So there's evidence of a divergence between the drag on a particle measured at the macro scale and the nanoscale. And this is really the idea behind this is a breakdown of the so-called Stokes-Einstein equation for the diffusivity of a particle. And some of the early reports are from not too far away. This is from the University of South Carolina, where they were looking at rotational diffusion of anthracene in polymer melts. This is polyisobutylene, polyethylene, and polydimethylsiloxane. The x-axis here is the molecular weight of the polymer. The y-axis is the viscosity. The open symbols are the viscosities measured using a rheometer, so it's a macro scale measurement of the viscosity. And the closed symbols are the viscosity measured by measuring the rotational diffusivity and using the Stokes-Einstein equation to calculate an effective viscosity felt by the molecule. So if you look at all of the plots, in all cases you see that at low molecular weights, there's very good agreement between the viscosity measured by rotational diffusion and the macro scale viscosity using a rheometer. But at a certain molecular weight, which is different for each of the polymers, and it's related to the number of bonds in the backbone, at a certain molecular weight, there's a divergence between the two. And this means that the Stokes-Einstein equation no longer accurately predicts the translational or rotational diffusion in this case of the molecule if you use the viscosity measured using a rheometer. In other words, even though it's a very viscous polymer, the molecule in this case is capable to rotate much faster than you would expect. And when I mean much faster, this is logarithmic. So we're talking orders of magnitude difference. For nanoparticles, a similar phenomenon was reported in 2007, where they looked at cadmium selenide nanoparticles in polymer melts using something called X-ray photocorrelation spectroscopy, basically dynamic light scattering with X-rays. You can do that at the argon light source. And what they did is they suspended particles in polystyrene of a single molecular weight, a high enough molecular weight, and they measured the diffusivity at 4 different temperatures. They measured the viscosity at that temperature, calculated the Stokes-Einstein diffusivity, measured the diffusivity, and they found that always the actual diffusivity was 2 orders of magnitude higher than the diffusivity calculated according to Stokes-Einstein. So again, the particles are capable of translating through the polymer matrix much faster than you would expect based on Stokes-Einstein and the viscosity of the melt. So again, a demonstration of breakdown of Stokes-Einstein. And the question is, why is this happening? So we got interested in this because, well, we thought maybe we can do this in a different way, maybe a novel way. And the way we do this is we take advantage of the fact that the particles respond to an oscillating field by rotating. And then we can measure that rotation magnetically. And the idea is you apply an oscillating magnetic field, shown in black, and you measure an oscillating magnetization, and you're going to have an in-phase component and out-of-phase component. And there are certain theories that you can use to interpret these measurements. And to make a long story short, there's a model by Debye that you can use that basically tells you that the in-phase component should be like the red line, the out-of-phase component should behave as a blue line, they should cross, there should be a peak, and at the peak, you have a condition that allows you to eventually determine the viscosity, okay? And some of the advantages of this is that you can use this technique with optically complex and opaque fluids, and the volumes this is a typo, this is microliters, are rather small. We've actually done measurements down to 20 microliters of fluid volume. And you don't need a lot of particles, so the concentrations of particles are below 0.1%. And I'm going to show you that this works. In terms of how versatile a technique this is, this basically shows you the peak frequency, location of the peak frequency as a function of medium viscosity for different particle sizes for commercially available instruments you can use to make these measurements. And the point I want to make here is that you can think of using probes of a wide variety of sizes between 10 nanometers and 1,000 nanometers. So you can really look at the probe size dependence of the mechanical properties of a complex fluid. You can make this quantitatively, and this shows work that we've done, reported in Soft Matter, where we use oleic acid-coated cobalt ferrite particles. The nice thing about cobalt ferrite is that at room temperature, it responds to an oscillating magnetic field by physically rotating. We determined the hydrodynamic size distribution in a reference fluid. In this case, it was hexane. We fit that to a log normal size distribution, and then we suspended the particles in a test fluid, in this case mineral oil, which we bought at a local pharmacy. And the idea was to see if we can determine the viscosity using this technique. This is an illustration of a asusceptibility spectrum for the particles, showing that it follows the Debye model. This is measurements of the out-of-phase component at different temperatures, showing that it shifts with temperature. And then you either estimate the viscosity from the peak, or you estimate it from a full nonlinear regression of the whole out-of-phase susceptibility plot or spectrum. And what you get are viscosities, in this case as a function of temperature. In black is the viscosity determined using a rheometer. In red is the viscosity determined from the peak position. And in blue is the viscosity determined from the regression. And in a fairly wide temperature range, the agreement is within 5%. And the reason why there's disagreement here is that for this sample, there was quite a bit of excess of free oleic acid, and at that temperature, it started to crystallize, and that led to some aggregation of the particles and the deviation in terms of the behavior. But the important point is that we can make quantitative measurements of the viscosity using this technique in simple fluids. I should have put this before, these particles are being made by the thermal decomposition method, and there are a variety of methods using acetylacetonates or pentacarbonyls. We use an iron oleate route. It results in hydrophobic oleic acid-coated particles, which are the ones that I used before, and we can make them with relatively narrow size distributions. For the other things that I'm going to show you, we want to coat them with a polymer that makes them hydrophilic, and we can suspend them in aqueous-based media. And in this case, what we do is we modify the surface first with a silane that has functional groups, and then we use these functional groups to react to another molecule, and the molecule can be either a dextran molecule or a polymer, in this case, polyethylene glycol. So with the polyethylene glycol particles, another example of this idea of making nanoscale and macroscale measurements is to use it to determine a so-called gelation temperature for a simple system that gels with temperature. In this case, I'm showing you data for solutions of gelatin, 10%. And you can measure gelation temperature rheometrically, or using viscosity measurements, by measuring viscosity as a function of temperature at a fixed shear, or by doing small amplitude oscillatory shear experiments and looking at where the in-phase and out-of-phase moduli cross. And in this way, I mean, I'm not going to go into all of the details, but the point is that you can determine a gelation temperature, say of about 25 degrees C using this technique, or about 28 degrees C using this technique, and you can see here that there's some hysteresis between gelation and melting. So what we did is we took our cobalt ferrite particles, coated with polyethylene glycol, so they don't have specific interactions with the gelatin molecules. This is at 70 degrees C, where the gelatin is completely melted, and you can see that there's an out-of-phase peak. Its location is a bit above our frequency range of our instrument. If we lower the temperature, we see the peak decreases in frequency, and then at very low temperatures where the matrix is completely gelled, we see that now we don't get an out-of-phase peak signal. If instead of doing the frequency measurements, we make measurements as a function of temperature, but at fixed frequencies, this is what we get. This is the in-phase component. This is the out-of-phase component. We start here with the gelatin completely melted, and then or dissociated, and then we lower temperature, and this is where we hit a transition where gelation is starting to happen. And we see a very sharp transition in the measurements, both in the in-phase and out-of-phase. And actually, if you look at the Debye model, you take advantage of the fact that the frequencies that we're using are rather low. You can expand the Debye model into something that looks like this. And when you're undergoing the gelation transition, what will happen is that the hydrodynamic drag on the particles is going to increase dramatically, very quickly. So what that means is that the relaxation time is also going to increase dramatically. And if you look at the Debye model, what that says is that when you hit the transition, this term here is going to increase very quickly with temperature, which means that this parameter here is going to decrease very quickly with temperature. On the other hand, when you look at the out-of-phase component, this term is going to increase very quickly with temperature. So that these measurements are consistent with the idea that the hydrodynamic drag on the particle is increasing dramatically as we cross this temperature. And the temperature determined from these measurements is about 21 degrees C, which is lower. This is the rheometer data, and this is the magnetic data, which is lower than the gelation temperature measured using a macro scale measurement. So you can superimpose the curves if you shift them, and there's a difference of about 3 to 4 degrees C in the gelation temperatures. What we think is going on here is that it's a probe size dependence. When you're at high temperatures, the gelatin is not associated to each other. It's free in solution. The particles can freely rotate, and the macroscopic viscosity is low. At a certain point, you start forming the initial parts of the gel matrix, but there's still enough space between the particles. So your rheometer starts to pick up these structures, and that can span the gap of the rheometer. But the mesh size, let's say, between the structures is still much greater than the diameter of the particles. So the particles can still freely rotate, and you only hinder the rotation of the particles when you've really gone below the transition temperature, and all of that gelatin has gelled, for lack of a better term. So I've shown you that it's quantitative. I've shown you that in gelling systems, there's deviations between macroscopic measurements and nano measurements. But the real interesting one is going back to the first things that I showed you with breakdown of Stokes-Einstein equation. So to test that idea, we used linear polyethylene glycol GPC standards of different molecular weights. This is the GPC traces. We measured the viscosities. They're all Newtonian. They are melts, but the molecular weight is low enough that the entanglements are not significant. There's not a lot of non-Newtonian behavior in these systems. And then we suspended PEG-coated cobalt ferrite particles. So the particles are coated with the same polymer as the melt. We measured the asusceptibility curves. We estimated the size in water. We fit the asusceptibility curves, and we determined the viscosity. And this is what we get. This is molecular weight of the polymer, viscosity in the y-axis. Again, it's logarithmic. 3 different temperatures. The closed symbols are the viscosities measured using a rheometer. The open symbols are the viscosities measured using the rotation of the particles. What we see, very similar to the measurements with anthracene molecules, is that below a certain molecular weight, there's very good agreement. The Stokes-Einstein equation applies. And above a certain molecular weight, there's a divergence between the nanoscale viscosity and the viscosity felt by a rheometer. And I don't have the time to go into the details, but an important point is that these are polymer grafted particles, and their hydrodynamic diameters are in the order of 25 nanometers. So the mechanisms that the prior work with cadmium selenide particles claimed would be responsible for the breakdown of the Stokes-Einstein equation are not applicable here. So there's something else going on, and we're actually looking to see what it is. My bet is that it's a de-wetting phenomenon because of the molecular weight of the melt exceeding the molecular weight, significantly exceeding the molecular weight of the grafted polymer. So that's it for the nanoscale viscosity measurements. Now I'm going to talk to you about the idea of using these particles to deliver thermal energy and using that, for example, to kill cancer cells. So the idea is quite simple. The idea is that it's commonly called magnetic fluid hyperthermia. You take particles, you target them to cells or to tissue, they internalize, you apply an oscillating magnetic field, the particles either physically rotate or the dipoles within the particles rotate. There's some irreversible energy loss there. Energy from the magnetic field is then transformed into thermal energy. It's delivered to the cell. The tissue temperature is taken to the hyperthermia range, and cancer cells are more susceptible to heat damage, and they die. I mean, that's the basic concept of it. Now, I've been showing this slide for a very long time. I think we first made this slide in 2004, 2005. And the first thing I always say, and the first thing that always comes to mind, is the idea that you can have targeted energy delivery at the nanoscale. I mean, that's really why this is attractive, the idea that you're making particles, and they can attach to internal organelles within the cell or to the membrane, or you can deliver them only to certain cells and not to others. And because the energy is being delivered in a targeted fashion at the nanoscale, perhaps that's more effective. And that's really what I want to talk about for the rest of the seminar. So the first thing is that particles dissipate heat. They do. If you apply an oscillating magnetic field of sufficient frequency and amplitude. And this is shown here. This is a simple illustration where we have a coil. We're putting a current through. We have a sample of magnetic particles suspended in a fluid. And within 15 minutes, the temperature can go from 23 degrees C to 66 degrees C. Now, a lot of papers in literature have plots that look like this one, where you see temperature starting at a certain value, and you apply the field, and the temperature rises to a certain number of degrees. And a lot of people make a lot of claims, and they report, oh, my particles raise the temperature of the sample by 60 degrees, so my particles are better at dissipating heat. And the first thing I want to point out that the temperature that's reached in this experiment is completely irrelevant, in the sense that it's really a function of the energy dissipation rate of the particles, but also how good a calorimeter or how good an experimental system you have. The more thermally isolated your sample is from the environment, the greater the temperature it is that you're going to reach. So what really matters is the initial rate of temperature rise. And the initial rate of temperature rise can be converted into a property of the particles that's called the specific absorption rate. And the way you do that is by multiplying by the heat capacity of the fluid divided by the mass of the particles. So that would be an intrinsic property of the particles. And it's going to depend, as you might imagine, on particle properties and on the properties of the applied magnetic field. And, there's a simple theory for this shown here, which basically illustrates that the energy dissipation rate is going to depend on the field amplitude and frequency, and it's going to depend on those are properties of the field, and it's going to depend on the so-called initial susceptibility and the relaxation time that I showed you before. So the initial susceptibility itself depends on the concentration of particles, it depends on the diameter of the particles, and it depends on the domain magnetization. So a lot of work in this field has been focused on changing the material so that we can tune the domain magnetization to make the particles more effective, or changing the diameter so we can increase the rate of energy dissipation. Now, when you change the diameter, you also change the relaxation times. And not only do you change the relaxation time, but as you might imagine, if you have a competition between two processes in parallel, you're going to have that the fastest process is going to dominate. So if your relaxation time, if your Brownian relaxation time is faster than your Néel relaxation time, you're going to respond to the field by the Brownian mechanism, and vice versa. So depending on the size of the particles and the size distribution, you could have particles that dissipate heat by either physical rotation or internal dissipation of heat. Okay, and that's an important point that I wanted to make. So the idea is we want to do this. The question is, how do we get the particles into the cell? And like with many nanomedicines for cancer, the real objective is to inject these particles systemically, make them small enough so that they extravasate in the tumor. They take advantage of the so-called enhanced permeation retention effect, and that would be a passive, let's say, targeting approach. And you also want to add molecules on the surface of the particles that attach to receptors and promote internalization, which would be an active targeting approach. And people have been talking about this for a long time. I've had a couple of conversations already in the last 2 days, which have been interesting. I'm not going to talk too much about this any further, but I'd be happy to discuss it later. So how do you engineer iron oxide particles for this application of hyperthermia? Well, the first thing is you need a core, an iron oxide or magnetic core. I prefer iron oxide because of biocompatibility. So the core is going to dissipate heat. You need a coating, and the coating is very important because it should provide colloidal stability, and most importantly, it should provide colloidal stability in biological fluids. And that has a lot of impact in our ability to deliver the particles to the EPR. And also, depending on whether the particles are colloidally stable or not, that can affect their toxicity, as we've seen and others have seen in experiments. You want them to be small so they can penetrate into the interstitial space of a tumor, but you also want them to remain small. You don't want them to absorb proteins. You don't want them to aggregate. And then you want molecules on the surface that allow you to target them to specific receptors. So the question is, can magnetic fluid hyperthermia, or this idea of heating, be applied to small tumors and metastatic cancers, to single cells, to subcellular structures? And really, the underlying question is there an advantage to delivering thermal energy using magnetic particles? And I'm focusing on magnetic particles, but similar questions would apply to other nanoparticle-based thermal therapies because some of the concepts are the same. So the way most people do these experiments, for example, in vitro, is they take cells, they incubate them with particles, they put them in a coil, they apply a magnetic field. Because you have so many particles in suspension, the temperature rises from physiologic 37 to about 43 degrees. You do this for a certain amount of time, and you measure viability. And that's really what most people do in vitro. And if you do that, these are controls. This is with 2 types of breast cancer cell lines, controls in the incubator, cells in the field with no particles, cells with particles and no field. And then these are the cells that had particles and field and therefore were subjected to hyperthermia. And you see that it's effective. You know, the particles themselves are not toxic, and cell viability is reduced in most cell lines. And the results seem promising, but they're promising in vivo only, in what I've seen, when you can inject the particles directly into the tumor. So delivery is a huge issue. As you probably know, clinically relevant cancers are most likely not accessible for direct injection, especially metastases. And the question still remains, if we're able to deliver particles to a small tumor or a small lesion of a small mass of cells, will we be able to heat them effectively? And I'm going to show you some more equations. And I'm an engineer, so I do have to have equations in my slides. Let's talk about energy transport and heat transfer in tissue and what it tells us about this idea of treating cancer with heating of magnetic particles. And to model this, I'm going to use the so-called Penn's bioheat equation. It's an equation that accounts for heat conduction in tissue, and also the idea that tissue is an effective medium with blood perfusing through. That blood can either deliver or carry away thermal energy depending on whether the tissue is hotter or colder than the arterial temperature. So we use this model. We consider a very simple situation, a spherical tumor with a uniform distribution of particles. Now, this is a highly idealized case. It's very unlikely that we're going to get a spherical tumor, and it's very difficult to get a uniform distribution. But this would be, in a certain sense, the best case scenario in terms of delivering particles to a tumor and achieving hyperthermia. So the question is, under the best case scenario, what does the theory tell us? We use tissue thermal transport parameters that are typical. We use typical particle parameters and typical magnetic field parameters, and we solve the equations. And I'm not going to show you the solution. I'm just going to show you the results. And if you have a 20-millimeter diameter tumor, the dotted line here would be the surface of my spherical tumor. And these lines are the temperature distributions. And what you see is that if you achieve a loading of about 0.6 milligrams per milliliter or milligrams per centimeter cubic, you don't get to the hyperthermia range. If you have about 1.2 milligrams per milliliter, then it seems like you're getting there. You really want high concentrations of particles to be able to heat the tumor. It seems like it's feasible. But if the tumor is 10 times smaller, let's say it's a 2 millimeter diameter tumor, it's a very small mass, something like metastatic cells in a lot of places in the body. To raise the tissue the temperature significantly, even getting into the hyperthermia range, you would need to go up to about 60 milligrams of core per cubic centimeter. And that is a serious, that is very difficult. That is a very serious obstacle. Getting enough particles to the tumor then becomes impossible. And therefore, this would seem to say that there's no advantage to heating. So as stated, the potential advantage, and I have to fix this, it's supposed to be MFH, is the nanoscale or intracellular delivery of heat to kill cancer cells. Steady-state heat conduction analyses, what I just showed you, indicate that the surface I didn't show you this, but it's a result of the analysis, that the surface temperature of a nanoparticle is practically the same as that of the surroundings. That a single cell, if we were to replace everything from inside the cell and just fill it with nanoparticles, the surface temperature of the cell would be no greater than 0.1 degrees C over the environment. And that there's a minimal tumor diameter that is treatable with this idea. So there doesn't appear, according to these arguments, no apparent advantage from nanoscale heat transfer. And this imposes serious limitations to this idea to treat small tumors in metastatic cancer. But an important point to make is that these are all theoretical arguments. And the real question is, what do the experiments say? And obviously, if the experiments didn't disagree with the theory, I wouldn't be here talking to you, I guess, right? So that's what I'm going to talk to you about. And I'm not going to be able to do a complete review. There's a lot more examples, and I'd be happy to talk to you about them. I'm going to talk to you about our own experiments. And then, can nanoscale thermal effects due to energy dissipation by magnetic particles lead to biologically relevant responses? And can they do so without a macroscopic temperature rise? And we've done a few things that I'm not going to show you, things like comparing external heating with heating by magnetic particles. And we always find that heating with magnetic particles is more effective at killing cells than external heating. But what I'm going to show you is only results where we don't get any heating, any temperature rise. So we do targeting. We decided to target cells through the epidermal growth factor receptor, which is overexpressed in many types of cancers. And there are various ways to target it. People use things like antibodies. In our case, just like many other people in the nanomedicine field, we were curious to see what would happen if we targeted the EGFR by using its main ligand, epidermal growth factor, which is a small protein. So our particles are coated with a polysaccharide carboxymethyldextran. They have EGF on the surface. The EGF is covalently bonded to the dextran. The dextran is covalently bonded to the particle, so they go with each other. They can't leave each other. And the experiments I'm going to show you first were with MCF7 cells that express a normal level of EGFR, and MDA MB468 cells, which express 100-fold more EGFR than MCF7. And I want to point out that there are some small differences in the sense that really high concentrations of EGF can lead to some proliferation and to some apoptosis, depending on the cell line. And I'm going to get back to that as I show you the data. So we make our particles. I'm not going to show you the details. We make our particles. And then the question is, does the targeting make a difference in terms of uptake by the particles by cells? We incubate the cells with particles, then we wash, we remove all of the unbound particles, we digest the cells using nitric acid, and we measure iron concentration using ICP-MS. And what we find, these are 3 different incubation times of 1 hour, 6 hours, and 10 hours. These are particles without EGF, particles with EGF. These are the cells that express a normal level of EGFR. These are the cells that express a higher level of EGFR. With the cells with a normal level of EGFR, we don't see any real, there's some differences, but not a significant difference in uptake between the non-targeted and the targeted cells. In the cells that do overexpress the EGFR, we see consistently, statistically significant increases in uptake of the EGF-coated particles in the cells for all incubation times. However, if you look at the amounts, the non-targeted cells, the particles, internalize in the order of 2 to 5 picograms of iron oxide per cell. And the targeted cells, do internalize somewhat higher in the non-targeted cells, which makes sense because, you know, they have EGFR anyway. The uptake is greater in the cells that overexpress the EGFR. But if you compare the baseline to the targeted, there's really not a dramatic difference. It's not orders of magnitude difference. However, it makes a difference in terms of where the particles go. And what we find, there's actually a time-lapse confocal microscopy imaging, particles in green, cell membrane in red, showing that the particles in the targeted cells, the particles with the EGF, are taken up quite rapidly. They accumulate within the cell, and there's a punctate pattern, which is consistent with going into endosomes and lysosomes. And actually using confocal microscopy with the cells that overexpress the receptor and cells that do not overexpress the receptor, we see consistent differences in that. In the cells that overexpress the receptor, we always see the green fluorescence associated with the particles, either associated with the membrane or associated with the lysosomes. It's kind of difficult to see, but we use an lysosome assay to sort of show that. In the cells that do not overexpress the receptor, we see some internalization. We see some punctate patterns that are consistent with endosomes and lysosomes. But we also see the particles in a lot of other places and not as well distributed. So the real question is whether it makes a difference in terms of killing cells, and specifically killing cells without a temperature rise. So the way we do this is we incubate the cells with the particles. We remove any unbound particles, and then we apply the magnetic field. And because you only have picograms of ion per cell, and you only have thousands, hundreds of thousands of cells, there's really not enough particles in the system to raise the temperature. So during the experiments, we monitored the temperature of the sample, and it always remained at 37 degrees C, just like our controls in the incubator. And then after treatment, we seed cells. 5 days later, we do an MTT assay. 10 days later, we did a clonogenic assay for the colony-forming ability of the cells after treatment. And this is the cells that do not overexpress the receptor. Again, 1 hour, 6 hours, and 10 hours of incubation time, and we have a incubator control. These are the particles with or without targeting in the incubator, no magnetic field. And then these are the particles with or without targeting in the magnetic field. We see cell viability decreases to about 75%, which is statistically significant decrease, but practically it's not really important because it wouldn't be enough for treatment. But the point is that these are the cells that do not overexpress receptor. Another important point is that the temperature did not change. So even though the targeting is not there or not as effective, there is a significant reduction in viability, just not practically significant. These are the cells that overexpress the receptor. And in this case, this is the incubator control. This is the particles in the incubator. These are the particles with the targeting in the incubator. And I told you that high concentrations of EGF can lead to apoptosis in this cell. So we're seeing some of that. We're definitely seeing some of that. These are the non-targeted particles in the field. We see that there's a more significant decrease in cell viability, but again, it's the 50% or so. And these are the particles that have the targeting in the applied magnetic field. And we see that in this case, cell viability after a single application of the magnetic field went down to about 4%. And again, the temperature remained constant. So one question is whether this reduction in viability correlates with the rate of energy dissipation of the particles. Everything I've shown you so far has the same, everything is with the same batch of particles. So the particles in all the experiments I've shown you, because the field conditions are the same, the rates of energy dissipation, whether there's targeting or not, are the same. We can change the rate of energy dissipation by changing the amplitude of the applied magnetic field. So, for example, low fields would give us a certain value, medium fields, another value, and high fields would give us a higher value. And I'm not going to go into the detail, they're technical, but the point is we do experiments with different field amplitudes, and that way we modulate the rate of energy dissipation. And if we do that, these are the cells that overexpress the receptor, 1 hour of incubation. This is the control, incubator control, non-targeted incubator control. These are the targeted particles without magnetic field. They again have some toxicity. Obviously, no field is being applied. It doesn't make a difference. These are the non-targeted cells with applied magnetic fields. At low fields, they barely change. There's no statistically significant change in viability, or actually surviving fraction using a clonogenic assay. As you increase the energy dissipation rate, you get a decrease in viability, but then if you increase the dissipation rate further, you don't get a further decrease in cell viability. On the other hand, with the targeted particles, what you see is a consistent systematic decrease in cell viability as the energy dissipation rate increases. And here we're killing 99-point-something% of the cells. So again, surviving factor depends on the energy dissipation rate of the particles, but still there's no temperature rise. So in every experiment I've shown you, the temperature of the suspension, of the cells, etc., measured with a fiber optic thermometer is 37 degrees C. So the question is, what is the mechanism? I mean, sure, we've seen these results. Why would this happen? So based on our confocal microscopy imaging, and because the particles are taken up by receptor-mediated cytosis, we hypothesized that it has something to do with lysosomes. So we wanted to look at the possibility of the particles, because of heating within the lysosome, because of energy being dissipated within the lysosomes, disrupting the lysosomal membrane and that causing cell death. So to look at that, we looked at an acridine orange assay. Acridine orange is a dye. It accumulates in lysosomes at fluorescence red. If you disrupt the lysosomes, then you would see a decrease in the red fluorescence of the cells. So this is flow cytometry, histograms, fluorescence intensity in the red channel, and count of cells. And this is the control, with and without. Black is without the magnetic field. Red is with the magnetic field. These are the cells that have not been dyed with acridine orange. Obviously, subjecting the cells to a magnetic field makes no difference in terms of the lysosomes. This is just free EGF, and also blue is my positive control, which is hydrogen peroxide, which is known to disrupt lysosomes. And we see in the blue a shift towards lower intensity in the fluorescence. These are the non-targeted particles. Applying the field definitely makes a difference. The red is shifted towards lower fluorescence. And then, sorry, these are the non-targeted particles. These are the targeted particles. And there you see a very significant decrease in fluorescence. So this is a single experiment. This is a summary of I think it's n equals 4. I'm not sure. It's in the paper. And shown as the fold change in the number of pale cells, meaning cells in this gated area, after applying the field relative to before applying the magnetic field. No change in the control, no change in the control where cells were subjected to EGF only. There's definitely an increase in the number of pale cells, so some of the lysosomes are being disrupted with the non-targeted particles, but there's a very significant increase in the number of pale cells, so we're really disrupting a lot of lysosomes with the targeted particles. The next question is whether the contents of the lysosome are being dumped into the cytoplasm. And for that, we looked at cathepsin B release from the lysosome. We use an assay kit that allows us to visualize cathepsin B. We use confocal microscopy, semi-quantitatively determine how much cathepsin B is in the cytoplasm. And this is a summary of measurements across a set of cells. And the important point is here, this is before applying the magnetic field. This is after applying the magnetic field. These are the cells that are, the particles that are targeted. And we see that there's a dramatic increase in the amount of cathepsin B in the cytoplasm. So this, along with the lysosome permeabilization measurements with acridine orange, seems to indicate that the particles are accumulating into the lysosomes, and then when you apply the magnetic field, they disrupt these lysosomes, dump the contents into the cell, and then they cause cell death. And one question is how, or what are some of the mechanisms involved? And one of the questions I get often when I talk about magnetic particles, because they're iron-based, is whether these particles catalyze Fenton reactions, which can lead to production of reactive oxygen species. So we measured reactive oxygen species, again, using a flow cytometry assay. I'm not going to walk you through the details, but the important points are the following. If we measure reactive oxygen species in the cells when they're subjected to EGF, non-targeted or targeted particles, and no magnetic field, relative to a control in the incubator, we find that the amount of ROS in the cells is practically the same as in the incubator. When we apply the magnetic field to cells in the absence of particles, there's of course no change. So this is full change in total ROS. There's no change in the amount of ROS. If we have free EGF and we apply a magnetic field, there's no change in ROS. Interestingly, when we have the non-targeted particles, even though we saw that they can disrupt the lysosomes, we don't see a significant change in the ROS production when we apply the magnetic field. But with the targeted particles, which lead to a lot more lysosomal disruption, we see that relative to the no field condition, when we apply the field, there's a significant 3- to 4-fold increase in reactive oxygen species production. And this is a positive control for ROS and adding an inhibitor. So we think this indicates that what's going on is that if you have particles that are not targeted or cells that do not overexpress a receptor, you get some particles attached to membranes, some particles internalized into lysosomes, some energy being delivered locally. That can lead to some disruption of lysosomes, but not enough to really damage the cell. But if you have loads, if you have targeting and you have overexpression of the receptor, and that leads to receptor-mediated endocytosis and accumulation in lysosomes, when you apply the magnetic field, you disrupt a large number of lysosomes. And the reason this is important is there's a growing interest in so-called lysosomal death pathways to attack cancer cells. The reasons being that lysosomal death pathways can be dependent and independent of apoptosis, can be effective in drug-resistant cells, and also the lysosome, production of lysosomes and production of proteases in lysosomes is upregulated in many types of cancers. It's associated with invasiveness of cancer. So we believe that perhaps this way we could target cancer cells and destroy them without a temperature rise. So an important point that I get asked a lot, especially from Sasha in the last couple of days, is whether local heating phenomena indeed exist. And I'm not going to show you all the data that we have because some of it is unpublished. I'll be happy to discuss it offline later. But I'll show you something we have published, and that is using a thermoresponsive fluorescent polymer coating the particle as a reporter of a thermally induced change on the surface of the particle. The polymer is polyisoproyl acrylamide, and the fluorophore is such that when the polymer undergoes a transition, the intensity of fluorescence increases. And the idea is we apply a magnetic field, the particles in the magnetic field dissipate heat, and we're going to monitor fluorescence as a way of measuring the thermal environment. I don't want it to say temperature, notice that. The thermal environment in the surroundings of the nanoparticle. This is fluorescent intensity using external heating, showing, and again, this is our first set of data, showing that below the transition temperature, which for some reason was quite high for this polynipam, we don't see much of a change in fluorescence, and then there's a temperature above which there's an increase in fluorescence, and then it plateaus again. Now, this is external heating. If we heat it using the magnetic particles, meaning that we have a fiber optic temperature probe, a fiber optic fluorescence probe in the magnetic field, we turn on the field when, at the dashed lines, this is time, this is intensity, which is shown in green, of fluorescence, and temperature shown in blue. What we see is that immediately upon turning on the field, the fluorescence begins to rise. And the initial temperature is about 20 degrees, which is much lower than the transition temperature for the fluorescence determined using external heating. So this seems to indicate that immediately upon turning on the field, something has happened in the vicinity of the nanoparticle, which has forced the polynipam to go from hydrophilic to hydrophobic. So it has crossed its lower critical solution temperature, let's say. So what do we call these phenomena when there are no macroscopic temperature rises? And some of us are starting to call it magnetically mediated energy delivery, the idea that you can use magnetic particles to transduce the energy of a magnetic field and transform it, whether into mechanical energy by pushing, pulling, rotating, shearing a structure, or into thermal energy locally. In all these cases that are shown here, magnetic particles do that. And in some cases, they do that without a macroscopic temperature rise. So this is a more general term, I think, than hyperthermia. And it's important because if we keep using the term hyperthermia to describe this field where we want to destroy cells by delivering thermal energy, we're always going to be confused as to whether a macroscopic temperature rise is important or not. And I believe that it's not necessary. To illustrate why this is important, what I did here is I calculated the concentration of particles needed to achieve the hyperthermia range as a function of the radius of the tumor for a variety of energy dissipation rates representative of what's in the literature. And the solid lines are the results of that calculation. And basically, the amount of particles needed decreases as you increase the amount of energy dissipation, but it always increases as the diameter of the tumor decreases. And this is because as the diameter of the tumor becomes smaller, volume to surface decreases, and therefore conduction away from the tumor becomes more effective than your rate of energy dissipation. Now, the dotted lines are a calculation of how much particles would be needed to deliver a similar thermal dose to the experiments that I just showed you that demonstrated we can kill cells. And the important point is that, yes, if the tumor is large, you need fewer particles to reach the hyperthermia range, but there's a critical size below which, and I'm not claiming to tell you exactly what that size is, but theoretically it should exist. below which it's more effective to take advantage of these nanoscale heating phenomena to kill cancer cells, in our opinion. So in summary, nanoscale, for this part, nanoscale heat delivery appears to be a promising approach to destroy cancer cells, even though heat conduction arguments indicate that there should be no local temperature rise. Experiments with cells and thermoresponsive polymers provide evidence of local thermal effects. And in my opinion, for many reasons, these observations may be due to energy flow in the vicinity of nanoparticles. This paradigm that you have to raise the temperature to hyperthermia range to kill cells may be wrong. Our results demonstrate that you can kill cells without temperature rise. And I think we need to take advantage of these nanoscale phenomena and understanding of the underlying mechanisms of cell death to design better particles to take advantage of these. And I think, we're hopeful, we're starting to do this in vivo now, that these methods could be used to destroy small cancer tumors and isolate cells. So I want to end by acknowledging this is my group now in Florida. I moved a year ago. So not all my group. I still have a few students finishing in Puerto Rico. I also want to acknowledge my collaborators in Puerto Rico, specifically Madeline Torres Lugo, who has been working on this project with me since we started, and funding from various agencies.